So without further ado, I think we should just get after it. And I would like to introduce Marco Vallejos. He was our summer intern this year, and he came to us from Williams College, where he just graduated with a degree in geoscience. And he's actually presenting remotely right now from up north. And he was with us for 10 weeks and just left this last week after he finished up his project because he was able to secure another internship with the NASA Development Program. So we are very, very excited that he is off on his scientific journey, and we look forward to hearing what he did this summer. And so his talk is titled, "Does Seagrass Influence Microplastic Retention in the Indian River Lagoon?" And I will let him take the stage.
Thank you, Lorraine. So I believe I'm screen sharing. And so I will be talking about haliduli radii effects on microplastics in the Indian River Lagoon, Florida. This was advised by Dr. Tilling at FOS, and I was the research intern this summer. So as we all know, plastic is one of the largest issues facing society, particularly in the oceans. It has become more and more evident that plastics are in the oceans, with the Pacific Gyre as one example. But another part of that pollution that goes very much unseen is something called microplastics. And while that has been popularized in the media, it also has a scientific quality to it. So I'm just going to go through that to kind of give us a baseline and then go into the project as we proceeded at FOS. So microplastics are less than 5 millimeters in size. They come from two sources. Primary sources are based in production, and those are nurdles and beads, which are used are melted down to create plastic resources.
Marco?
Yes?
Sorry to jump in on you. Would you mind doing it in the screen or in the slideshow mode so we can read your slides better?
Oh, sorry.
No problem. Thank you so much.
Sorry about that. So secondary sources come from macroplastics. So think water bottles and plastic bags in the ocean themselves. These form microplastics through degradation, usually through UV sources or wave forces, and create fibers or fragments and sometimes beads that are then seen in ecosystems. So that led us to want to look at how this affects seagrass habitat and if seagrass habitats are acting as a catchment for microplastics. So to go through some geophysics for why we are looking for microplastics in the seagrasses, wave orbitals are small circular forces that diminish down the water column, and if they come into contact with a seabed or a seafloor and their sediment to be suspended, that sediment usually will suspend. And so then with that, seagrasses and other types of barriers such as reefs or mangroves, as an example, can reduce that energy, which would then lower the energy itself in the system and then cause sediment to fall out of suspension. And researchers have found that microplastics also seem to behave like sediments similar in size. And seagrasses act as a critical habitat for fauna in the area, usually as a nursery and foraging area. And we wanted to look at whether or not haliduli radii is acting as a catchment, as other research has shown that other seagrass species have been found to have microplastics on them. So to reiterate, do haliduli radii beds act as an effective catchment for microplastics was our main question. We also wanted to see whether or not these microplastics settled on the blades or if they settled out of the water column into the habitat around. So I'll go into the variables that we looked at. And we also wanted to see if there was a difference in microplastics accumulation between the seagrass habitat that we looked at or bear habitat. So our experimental design is as follows. We sampled in one day in August, and we had 6 replicates for each habitat. So that being the seagrass habitat and the bear habitat. Of our surface water, sediment surface water, and sediment core variables, we took 2 subsamples. And with seagrass blades in the seagrass habitat plots, we did 4 subsamples. And on those plots themselves, they were 1 meter by 1 meter. We also did field measurements to calculate percent occurrence, percent abundance, seagrass type, and canopy height as well. And so with lab methods, I'm just going to go through an overview because we had to analyze each or prepare each variable in a unique way, which then led to an associated water sample that we assumed would contain microplastics, which we then vacuum filtered onto a 45 micron paper filter to allow us to begin analysis and for microplastics, to look for microplastics. So with seagrass blades, we collected the blades in filtered salt water in vials, which we then transferred to a collection jar, and then filled that jar with 50 milliliters of filtered water, and then agitated that jar itself for 2 minutes to remove any microplastics and epibionts on the blades themselves. After that 2 minutes, we removed the seagrass blade and then documented each blade in each side, as well as scraped those blades, and then to remove any potential epibionts and microplastics still on the blade itself, which we then, and all those tools that we used were then rinsed into the associated jar, which we would then filter for each seagrass blade. With sediment cores, these cores were 5 centimeters at minimum. Most of them were greater just due to sampling technique. And we would dry those samples in a weigh boat by tapping out the core and then dry it in a 50 centigrade oven, which would then allow us to rehydrate the sediment in a hypersaline solution that was of 250 grams per liter of table salt. This would then be agitated to rehydrate the sediment, and because of the hypersaline solution, the microplastics should float out of the and separate from the sediment, which then allowed us to pipette off the top 200 milliliters of water to filter and find microplastics. Water samples were done with the same process, as both were in 1 liter Nalgene bottles. These Nalgene bottles and all equipment was triple rinsed before any analysis was done in order to minimize contamination. Both water samples, surface water and sediment surface water, were then allowed to rest to allow any suspended solids to settle out of solution. And then we would do a partial pour of each bottle to filter said bottle. And usually it averaged out to have a remaining 100 milliliters of water left in the bottle, which was then discarded. And this allowed us to have clearer filters, which were made for easier analysis. So for the identification, we followed the Marine and Environmental Research Institute Guide to Microplastic Identification. This showed a reading procedure, common occurrences, rules, and exceptions to plastics and what they may look like and how they may behave under certain stressors, which were heat tests and as a flame or a needle. It also identified types, colors, sources, and shapes. For our study, we almost exclusively found fibers in all samples, and we had to eliminate clear fibers from our study due to a contamination source that was discovered. So here's the data, and the next 3 slides will look the same. All axes will be the same. The only thing that will be changing will be the variable that is being looked at. So for this, we have surface water. which on the left side is number of microplastics, and then on the right side will show length in millimeters. And so as you can see, we found that the numbers were fairly low in both habitats on average, and the sizes look to be greater in the seagrass habitat and less in the bear. However, due to the high p-value, we have to understand that it's not as statistically significant. Sediment surface water shows a reverse trend, or a same trend in the number of microplastics, where seagrass has less than the bear plots. However, in length, it showed that they were shorter than the bear plots themselves. Again, notice the p-values. These seem to not have statistical significance. Finally, in sediment cores, we showed the reverse trend in numbers, where seagrass showed that there were a higher number of microplastics found in the sediment core than in the bear habitat, while the length was about equal. Again, p-values are very high. So this didn't show much difference in terms of both habitats and which microplastics were accumulating and found in those habitats. However, within the seagrass habitat, we saw an important change, which also was statistically significant, in that a majority of the microplastics within the habitat were found on the seagrass blades themselves. Size was less significant. However, this allows us to conclude that the seagrass blades, the Halidrili redii seagrass blades, are in fact capturing the microplastics that are in the water column that flow through the seagrass habitat. So that leaves us with a few conclusions. No significant difference was found between the Halidrili redii seagrass plots as compared to the bear plots. Also important to note is that these plots were less than 10% density of the seagrass. So that would be a study to show, in a larger study, would be to find seagrass plots that are more dense to see if that is causing a change in how microplastics accumulate in the habitat as compared to a bear habitat. What we did see was that Halidrili redii blades do have an effect on the distribution of microplastics in the water column, where the seagrass blades themselves are capturing the microplastics rather than acting as a sink as we originally expected, and as was seen in other studies. Again, due to our density, this would potentially change as the density increases. Future studies would lead to a larger scale study of the distribution of microplastics as in seagrass habitats as compared to bear habitats, and also look at the ecological effects of microplastics in seagrass habitats, as they are known to cause issues with megafauna. And so this could allow us to look at other types of ecosystem effects from microplastics. So finally, I'd like to acknowledge the Florida Oceanographic Society's research department. Thank you for everything that you have done. Thank you to Katie, particularly for advising this project, and for Sam for being in lab all the time with me. Special thanks to Haley Miller, who was the shadow who helped us with sample collection. And a final thank you goes to the FOS staff and the Coastal Center for being my volunteer home for this summer. And then now I will look at questions. So for Jessica, so that goes with our filtration, with our triple rinsing. Nalgene bottles are expected to not break down from what we were. Yes.
Can you read the question aloud? No one else can see the question that was posted. Thank you.
Sorry. So Jessica Frost asked, wouldn't using sterilized glass sample bottles be better to use and eliminate the potential for plastic contamination for the container itself. So yes, with the Seagrass vials, they were glass. However, they did have a bit of a rubber stopper on top, which could potentially cause contamination. Same goes for the jars that we used. They had a rubber stopper. They were glass. That likely caused the contamination of clear plastics. However, colored plastics were able to be associated with the field samples themselves. And then a follow-up from Jessica. Was there a control of just the nalgene sample bottle? Yes. So we did use controls, procedural blanks for each sample. Okay. Susan asks, what eats the seagrass that would be ingesting the microplastics? Good question. I am not sure. That is not my field. However, I'm sure Katie or Lorraine or many others could answer that. Agitation was performed by shaking the bottle for 2 minutes and was expected to be, agitation was done just kind of by shaking a mason jar for 2 minutes, and we couldn't really uniformly do that any other way of me trying my hardest to be the same for each. Vincent asks, yes, we only, he asked if only shoalgrass was examined. We only examined shoalgrass for this particular study. However, I imagine that including other seagrass species could potentially lead to differences due to the size of the blade and the size both in length and width, and so general surface area. Was the seagrass sampled in the nursery or game fish lagoon? The seagrass was sampled in the right off the FOS Coastal Center property. So right where the dock is. And yes, the graduate reference was included. What was the, I don't know that word, epiphytic biomass from the blades? Jessica Frost asked this. Wouldn't the bacterial film from these epiphytes be attributed to adhering to microplastics rather than the blades themselves? Oh, okay. So the research has shown that yes, the epibionts are part of what is adhering to, as what is capturing the microplastics that are then on the blades themselves. And so that was what we did through the study. To calculate biomass, I'm not sure how we would have done that for this pilot study, but I imagine that would be something that would be included if this study were to be expanded. Susan asked where the samples were taken. Right, I failed to include that. They were taken off the FOS property in the Indian River Lagoon. Dominant color, Jerry Snyder asked, was there a dominant color of fibers? I don't have a number, a quantitative number. Qualitatively, light blue and blue seem to be the most frequent from just my memory of doing this study. Anissa Dial asks, do you think you will continue working on the relationship that seagrass and microplastic in the future? It isn't the plan for FOS to do that. I hope to be one day, but for now I'm back in Massachusetts, very far from the ocean. Excuse me. Thank you, Vincent. Your donation would be greatly appreciated. Amanda Khan asks, could you please explain the difference between the second figure on the last graphic slide, Haladuli water, and water over seagrass bed on the previous slide? Could you please explain? So I think this is the slide you want me to look at, Amanda. The difference with the second figure shows here that it's just to do with length. And I think, I can't see the p-value, but I don't think it was significant. So that I kind of skipped over that, my apologies. The length there, we couldn't really find anything that statistically showed evidence of the size varying too much within the different variables. So I can't answer that any more than that. Amanda also asked, was blade density among replicate sites similar? Was it quantified? It was quantified. It was less than 10% for each plot, and I don't believe it varied too much. I would have to look at the data one more time, but it usually was kind of in the 2% to 8% value. And then Jerry asked, does the blue color suggest a temporary roof tarp source? We could not analyze for source because that would require some sort of chemical digestion. So I can't answer that. And that is currently all the questions that are in. So if that does it, then I think I am done and can transfer it to Lorraine or I think Sam. So thank you.